The present topic of discussion, it is on cell injury, cell death and cellular adaptations, which is the second part. So here we will be discussing about irreversible cell injury in detail. That is irreversible cell injury types, that is the cell death types like necrosis and apoptosis will be dealt in detail. Now let's see about irreversible cell injury. So basically how does the pathogenesis occur? So what happens before this irreversible cell injury, that we will see first. So as we have discussed in the first part, a normal cell, it will maintain homeostasis, but whenever there is a stress, it will try to undergo adaptations, that is cellular adaptations. Whenever it is unable to adapt to it, or whenever the injurious stimulus is more, it results in cell injury. And this cell injury is of two types, that is reversible and irreversible, and depending upon the intensity of the stress. If it is a mild stress, it results in reversible, but if it is severe and progressive stress or progressive injury, it results in irreversible cell injury, and that it means that it is a cell death which is in the form of either necrosis or apoptosis. So this is about irreversible cell injury, wherein the injury is more severe as well as progressive. Okay, so what are all different causes of this cell injury? So there are different causes of cell injury. First of it is hypoxia and ischemia, wherein there will be decreased blood supply, because of which there will be lack of oxygen to the tissues. Because of deprived oxygen, there will be injury to the cell. And next is chemical agents. So there are various chemical agents and drugs like environmental pollutants, insecticides, pesticides, cyanide, arsenic, mercury. These are all various chemical poisons which can result in cellular injury. And even physical agents are there like mechanical trauma, radiation, electrical shock, thermal trauma, any changes in the atmospheric pressure, all can result in cell injury. Okay, and there are some other agents like microbial agents or infectious agents in the form of bacteria, viruses, fungi and parasites, all also can result in cell injury. So these are infectious agents, and sometimes even immunological reactions which occurs in the body. Okay, so which actually are good for the body as well as bad. So some immunological reactions like hypersensitivity reactions, that is anaphylactic reactions, and some autoimmune diseases, they can result in cell injury too. And sometimes even nutritional imbalance or nutritional derangements like protein energy malnutritions like kwashiorkor or marasmus, and sometimes even starvation can result in cell injury. And some genetic disorders also, okay, that can result in cell injury. And last but not least, it is aging. Any cellular aging or senescence, it results in decreased replication of the cell and decreased repair of the cell. So as there is no replication and repair, those cells also will undergo cell injury. So these are the various causes. So now let's see the types of these cell death. That is, first of it is necrosis, which is a type of irreversible cell injury. So now let's see how necrosis occur. Actually we have discussed about some of it in our first part, but let's see how in necrosis it will act. So if you see, there is decreased oxygen supply, toxins and radiation. So these are all different causes of cell injury which can result in mitochondrial damage. So whenever there is mitochondrial damage, as mitochondria's function is oxidative phosphorylation, right? So there will be decreased oxygen for oxidative phosphorylation, which results in decreased ATP generation. So whenever there is decreased ATP generation, there will be different consequences like a sodium pump will be defective, glycolysis will be anaerobic glycolysis, and sometimes protein synthesis will be defective, resulting in cellular abnormalities. And also there will be increased production of reactive oxygen species. That means there will be production of increased free radicals which are harmful for the body. So this all happens because of the injurious agents. So what happens because of the increased reactive oxygen species? So there are various pathologic effects like lipid peroxidation, which can result in membrane damage, as our cellular membranes are made of phospholipids. And sometimes there will be protein breakdown and protein misfolding. And sometimes it can result in damage to the DNA, which can result in mutations and sometimes causes tumors. So this decreased ATP generation and increased reactive oxygen species causes some cellular abnormalities which can result in necrosis. Now let's see what are all the cellular abnormalities that occur in necrosis. So if you see this next picture, so the cellular abnormalities will be in the form of, as you can see, membrane is broken down and the contents are leaking out. So there will be breakdown of plasma membrane and there will be leakage of the contents into the extracellular matrix. Whenever there is leakage of the contents towards outside, it can result in inflammatory reactions. So too much of inflammatory cells will come into the action. Like our first acute inflammatory cells will come, later followed by chronic inflammatory cells. And after inflammation there will be occurrence of myelin figures. These myelin figures are nothing but these are some aggregates of damaged cell membranes which are seen more in case of necrosis compared to reversible cell injury. Okay, and sometimes there can be dilation of mitochondria in the form of amorphous densities and disruption of lysosomes can be there. Okay, all these changes can be seen within the cytoplasm of a cell in necrosis. So what are all the nuclear changes? Now that we have seen cellular changes, right? Cytoplasmic changes. So if you see here, there are 4 different pictures are there, and the first picture represents a normal cell with a normal nucleus. Okay. Whereas if you see the other 3 types, that is the first one is pycnosis, where there is a shrinkage of the cell with condensed nuclei and increased basophilia also is there, that is pycnosis. And karyohexis means there is fragmentation of the nucleus will be seen. Okay, nucleus will be fragmented. And then the third pattern is karyolysis, wherein the lysis of the nucleus will be there, that is basophilia of the chromatin will fade off. So these are all the 3 types, that is pycnosis, karyohexis and karyolysis are the nuclear changes which are seen in a cell with necrosis. And there are different types of necrosis that can occur in different cells and different reactions, like coagulative necrosis, liquefaction necrosis, caseous necrosis, fat necrosis and fibrinoid necrosis. Now let's see about each type of necrosis in detail. First is coagulative necrosis, and it is the most commonest of the necrosis patterns that can occur. So this coagulative necrosis usually occurs because of 2 factors, that is because of ischemia, that is lack of blood supply, and also sometimes because of the bacterial and the chemical agents the coagulative necrosis can occur. So how does this appears under a microscope? So if you see this microscopic picture, it is a picture of infarct kidney. So if you see, there are 2 parts in this. One side, on the left side, it is a normal kidney epithelial cells, and on the right side it is infarct kidney, that is necrosis. So if you see there, the cells, the necrotic cells have preserved cellular outlines, but you cannot see any nuclei as you can see on the normal side, and you can see some blue dots between the tubules. They are nothing but inflammation. Okay, so what happens in coagulative necrosis? The basic cellular outline is maintained in the form of tombstones, but the cytoplasmic and the nuclear details will be lost. Okay, as you cannot see the nucleus, right? And inflammatory cells will be seen in between these tubular epithelial cells. Okay, and these necrosed cells will be swollen and they appear more eosinophilic compared to that of the normal cells. As you can compare in the picture only, that the necrosed cells are more swollen and they appear more pinkish, that is more eosinophilic in nature. But why these changes are occurring in coagulative necrosis is because these changes are because of the denaturation of cellular proteins as well as enzymatic digestions of the cell. And this coagulative necrosis occurs in all the solid organs except for brain. Okay, so this is about coagulative necrosis. Next we shall see about liquefaction necrosis, which is also called as colliquative necrosis. And here this liquefaction necrosis, it is also because of ischemic injury as well as bacterial and fungal infections. So what happens in liquefaction necrosis is that this ischemic injury and bacterial fungal infections, they release hydrolytic enzymes. So what these hydrolytic enzymes will do is like they cause tissue degradation resulting in semi-fluid material. That is, the dead cells will be completely digested, transforming the tissue into liquid mass, liquid viscous mass. Okay, and this liquefaction necrosis is seen in infarct brain and abscess cavity, whereas coagulative is seen in all other solid organs except brain, whereas liquefaction necrosis it is seen in infarct brain. And how does it appear microscopically? So as dead tissue is there, abscess cavity, right? So there will be a cystic space which will be formed, which will be filled with necrotic cell debris and macrophages. So those are the inflammatory cells which are seen here. And because it is in the brain, right? So there will be proliferation of glial cells will be there, and also proliferation of the blood vessels can be seen in liquefaction necrosis. And the next type is caseous necrosis. It is one interesting variant, and this caseous necrosis actually the term caseous means cheese like, and it is seen in tuberculous infections. If you see, this is a tuberculous infection where that is all is that yellowish cheesy material. So because of that gross appearance of that yellowish white material, right, so that name is given as caseous necrosis. As I said, it is seen in tuberculous infection. So how does it appear microscopically? So if you see, this is a microscopic appearance of a caseous necrosis. So in the center there is some pink material, right? That is nothing but caseous necrosis, which is structureless eosinophilic material, and if you give a closer look, it will have that granular debris. It will be in granular pattern, and surrounding this caseous necrosis will be granulomatous inflammation in the form of, as you can see, those white areas, light pinkish areas, those are nothing but population of epithelioid cells which are modified macrophages. And you can see those blue dots, right? Those are nothing but a population of lymphocytes. Okay, and surrounding this we see different types of giant cells, that is either Langhans giant cell we will see, or foreign body giant cells we will see. So all this together is called as a granuloma. So what all we will see in caseous necrosis now? Necrosis in the center, which will be in the form of structureless eosinophilic and granular material, surrounded by epithelioid cells which are modified macrophages, and then a population of lymphocytes. And then we will see both the Langhans and foreign body giant cells, which altogether is called as a granuloma. So this is about caseous necrosis. Now let's see fat necrosis. So this is also a special form of cell death wherein it occurs in fat rich anatomic locations like pancreas. Okay, so here because of the necrosis there will be release of pancreatic lipases which are released by injured tissue, that results in necrosis of this pancreatic tissue as well as fat deposits. So how does this fat necrosis, right? So here what happens is that there will be hydrolysis as well as rupture of this adipose tissue. Whenever there is rupture of adipose tissue, right, this neutral fat will be released. That released neutral fat will change into glycerol and free fatty acids. And these fatty acids which are released, they form a complex with the calcium which is present outside, resulting in the formation of calcium soaps, which is saponification, that we will see as chalky white areas. So this is fat necrosis. And if you see microscopically, as it occurs in adipose tissue, right? So here if you see, there will be cloudy appearance of this necrosed fat cells. Okay, and this necrosed fat cell, that is cloudy appearing necrosed fat cells, will be surrounded by mixed inflammatory cells. So that is how the microscopic appearance of fat necrosis will be. Next is fibrinoid necrosis. So this is also a special form of necrosis. Actually here this is seen in immune reactions. So any immune reaction is a immune complex reaction, is an antigen antibody reaction, right? So here what happens is that an antigen and antibody complex along with fibrin, it is seen in the walls of the arteries. So whenever this complex which is seen in the walls of the arteries, when it is leaked out of the vessels, it gets deposited on the surface. Okay, resulting or giving an appearance of bright pink and amorphous appearance. So this bright pink amorphous appearance of this complex is called as fibrinoid. So it is basically an immune complex reaction. So if you see this picture, microscopic picture, that center round one, that whole thing is the wall of an artery, and that pink amorphous appearance, that is called as fibrinoid, and the surrounding the fibrinoid is dense population of inflammatory cells. So this is how a fibrinoid necrosis will look like. So with this, necrosis is over. Next we will move into another type of irreversible cell injury, which is nothing but apoptosis, where it is a tightly regulated intracellular program. So as necrosis, it is commonly seen in pathological events, right? But whereas apoptosis is seen even in physiological conditions also. So apoptosis is seen in physiological, adaptive as well as pathologic events. That is why apoptosis is termed as coordinated and internally programmed cell death, so which serves to eliminate unwanted and potentially harmful cells. So how does an apoptosis occur? That is, what is the mechanism of apoptosis? So it is actually by activation of some enzymes called as caspases. So these caspases can be activated or inactivated with the help of pro and anti-apoptotic proteins. So in the process of activation of these caspases, basically we have two types of caspases, that is initiation caspase and execution caspase. Depending upon that only we have two phases. So first is initiation phase, wherein there will usually these caspases, they are present in inactive form in the cell. So they have to be turned to active form. So in the initiation phase, there is activation of initiator caspases. The commonest ones are caspase 8 and 9. Whereas in executionary phase, there will be activation of executioner caspases. Okay. The commonest ones are caspase 3 and caspase 6. So this initiation phase, that is activation of caspase 8 and 9, it can occur in two pathways. That is, it can occur in extrinsic pathway, that is by signaling from outside, or as an intrinsic pathway it can occur. Okay, now let's see initiation phase followed by execution phase. So that is, first we will see extrinsic pathway. This extrinsic pathway, it is also called as death receptor initiated pathway also. So if you see, this occurs on the cell surface. So if you see, this is a cell with a cell membrane, where on the surface of the cell membrane we have some proteins called as Fas proteins. So these Fas proteins are nothing but death receptors, which belong to a family called as tumor necrosis factor receptor family. So these are basically death receptor family. So normally Fas is present as an inactive form on the cell surface. So it needs a ligand to come and bind to it to activate the Fas. So now a ligand has come and bound to this Fas, which is nothing but Fas ligand, Fas L. So this activated Fas ligand, it brings 3 or more molecules of the Fas together. It results in the activation of the cytoplasmic death domain. So in the cytoplasm of the cell, there will be death domains. So these blue ones are the death domains which are activated now. So these death domains are called as Fas associated death domains, FADD. So these Fas associated death domains, they will convert the procaspase 8 to active caspase 8. So this procaspase 8 is an inactive form of caspase 8. So FADD will convert the inactive caspase to active caspase 8. So this is an initiation pathway. This is an initiation phase in extrinsic pathway. Now let's see intrinsic pathway. So this occurs in mitochondria. So that is why it is also called as mitochondrial pathway. So here what happens, between the cell membranes of this mitochondria, there will be a protein called as cytochrome C, which will be tightly regulated only inside the mitochondria. It will not be moved outside. But whenever our cell, right, it loses its cell survival signals, okay, that its survival is waste. So that kind of signals, so loss of cell survival signals, then the permeability of the mitochondria will increase. So whenever the permeability increases, this cytochrome C, which is bound inside the mitochondrial membrane, it will move outside into the cytosol. So in the cytosol, this cytochrome C, it will go and bind with a protein called as EPAF1, which is nothing but apoptosis activating factor 1. So this complex of cytochrome C and APAF1, they will convert the inactive form of caspase 9, that is procaspase 9 to caspase 9, which is an initiator caspase. Okay, so this is when there is loss of cell survival signals. But what happens when there are good signals to the cell, that is good growth signals, that means to regulate the apoptosis. So whenever there are growth signals, there is a protein called as BCL2 or BCLX. These belong to a family of BCL2. Okay, B cell lymphoma proteins. Okay, this BCL2 or BCLX has a tendency to stop the movement of this cytochrome C from mitochondria to the cytosol. Like that, they will stop the mechanism of apoptosis. That means BCL2 and BCLX are the ones which are anti-apoptotic proteins. So now we got to know that caspase 8 in extrinsic pathway and caspase 9 in intrinsic pathway are the initiator caspases. So this is about initiation phase. Now let's move into execution phase. So here if you see, this is a cell wherein inside the cell these initiator caspases which are activated, right? Now they will activate the executioner caspases. That means previously I told you executioner caspases are caspase 3 and caspase 6. So these procaspases 3 and 6 which are present in inactive form are converted to active form, that is caspase 3 and 6 by initiator caspases. So what these executioner caspases will do is they help in changes in the cell, that is changes in the nucleus as well as in the cell. So they cause endonuclease activation which results in changes in the nucleus by forming a condensed chromatin, and sometimes it can also result in fragmentation of the nucleus also. And the second mechanism is that it breaks down the cytoskeleton. So whenever cytoskeleton of the cell is broken down, the cell loses its shape and it starts to form the cytoplasmic blebbing, and also it results in the formation of it breaks down and forms apoptotic bodies. Whenever these apoptotic bodies are formed, right, the phosphatidylserine, whatever is present on the inner side of the cell membrane will move towards the outer side. So those will act as ligands for the phagocytes. So now that phagocytes has seen its ligand, so they will come and they will start to phagocytose the apoptotic bodies. And here when compared to necrosis, in apoptosis there will not be any acute inflammatory reaction. Why? Because there is no leakage of the cellular contents towards outside. That is the main difference. There will not be any inflammation. So now let's see about apoptosis in biological processes. How does apoptosis occur in biological? As I told you, apoptosis even occur in physiological conditions as well as in the pathological conditions. So in physiological conditions, that is the conditions where apoptosis occur is during embryogenesis. So like not all cells during embryogenesis will be seen in the final infant or final fetus. Okay, so there will be some programmed destruction of the cells during embryogenesis of few cells. And sometimes there will be involution of cells in hormone dependent tissues like endometrial shedding will be there, lactating breast regression will be there after breastfeeding, and sometimes there will be cell loss in proliferative cell population, and thymus will be involuted in early age, and also sometimes there is elimination of harmful self-reactive lymphocytes. If these self-reactive lymphocytes are not eliminated by apoptosis, they result in autoimmune diseases. So that is why apoptosis is very important in physiological conditions. So now apoptosis in pathological conditions, so where all it will act. So some conditions wherein any physical, chemical, nutritional, thermal injuries like radiation or cytotoxic anti-cancer drugs or extremes of temperature, those tissues which are exposed to these things, so those tissues can undergo apoptosis, and sometimes accumulated misfolded proteins can undergo apoptosis, and sometimes there can be cell death or cell injury in some viral infections, and in case of pathologic atrophy of organs and tissues on withdrawal of stimuli, something like prostatic atrophy and all, so where there will be apoptosis which is seen. So like this is about apoptosis in the biological process, that is both in physiological as well as in pathological conditions. But how do we check out for apoptosis, that is how do we demonstrate apoptosis basically. So there are some special stains and special techniques by which we can see apoptosis in tissues, that is by chromatin condensation staining by use of hematoxylin stain and fuelgen stain, and some fluorescent stains with acrylic orange dye, and sometimes techniques like flow cytometry can be used to visualize rapid cell shrinkage. And these DNA changes which are seen in apoptosis can be seen by in situ techniques as well as gel electrophoresis. And as I told you earlier, this phosphatidyl serine will be seen towards the exterior, right? So this phosphatidyl serine can be stained by using IHC stain with annexin 5. Okay, so these are all various ways by which we can demonstrate apoptosis in a cell. So this is about cell injury, cell death as well as cellular adaptation, second part, wherein we have discussed about irreversible cell injury in detail, its causative factors and necrosis types, and apoptosis and of which mechanism of apoptosis we have discussed and its role in biological processes. So both necrosis and apoptosis are very important if you see in exam point of view, both as essays as well as for short notes.